So what we will see today together, it is a specific application of computational toxicology, a specific application of computational toxicology. Computational toxicology is a discipline that using existing information tries to build mathematical models able to predict properties and, in our case, toxicological properties of molecules. And the applications of computational toxicology are many, and they range from risk prioritization to hazard assessment of existing chemicals or new entities, new structures. The applications that we wanted to focus on today, it is the assessment of the potential mutagenicity of impurities in pharmaceuticals. And the aim, it is to limit the carcinogenic risk associated with the presence of these impurities in our drugs. So in this specific application, the property we are trying to predict, it is very well defined, and it is that the mutagenicity of chemicals. And typically, this type of mutagenicity that we are trying to monitor, it is related to a reaction between the chemical and the DNA. So it is well defined and well described. This type of mutagenicity is detected in vitro or in vivo by a variety of different tests, different assays. And the assay that it is typically used for mutagenicity in vitro, and it is the assay that is described in the guidelines, is called the bacterial reverse mutation assay, or the Ames test. So the Ames test is an in vitro assay that is able to detect the mutagenicity of chemicals. Now, the in silico assessment that is described in the guidelines requires the use of two different methodologies that has to be used together. One, it is an expert rule-based methodology, and the other one is statistical-based. And today together, we will see what they are and why, using these two methodologies together, you can improve the quality of the prediction and the reliability of your prediction. This concept, the concept that you can build a relationship between a structure and a property of chemicals is not new. And above all, related to mutagenicity, we can trace back to 1988, one of the first publications by Ashby and Tennant, that gives a list of fragments that were known to be related to genotoxicity and mutagenicity. And this is the macromolecule that they drew, including all these different functional groups. In this case, the relationship between the structure and the activity has been built by human experts. But nowadays, we use a computer to build this relationship, and then this relationship is encoded in a software that is able to give you the prediction. So the idea is to build a connection, a relationship between the chemical structure here and a measure activity, a measure property. The issue is that the chemical structure is quite a complex entity, and so it is impossible to build a direct relationship, a direct model between this complex entity that is the chemical structure and your measure property. What you have to do first, it is to translate this object into a mathematical representation, and then you can build the relationship, the model between the mathematical representation of the chemical structures and your property, the mutagenicity. Now, according to the type of mathematical representation that you use, you can develop different types of models. If you use as a mathematical representation the 2D structure, you build rule-based models. Instead, if you use what are called the molecular descriptors, you can build the so-called statistical-based models. Molecular descriptors are a way to describe these complex objects, and there exist thousands of different types of molecular descriptors. So according to the description of the molecule that you choose, you can develop either a rule-based or a statistical-based models using statistics. The rule-based models are based on what it is called a knowledge base, and the knowledge base, in the knowledge base you have chemicals with their activity. So this is an example. We have different chemicals. Some of them will be Ames positive, some will be Ames negative. And with statistical approaches, what you do is that you try to group chemicals that have the same type of activity. So you try to group chemicals that are Ames positive. And these chemicals must be similar. And once you've done similar groups of chemicals, you try to understand which is the chemical feature that gives that is common to all the chemicals, and these are the responsible for the mutagenicity. In this case, it is the epoxide. And this epoxide is the so-called structural alert, and it is the feature that is responsible for the mutagenicity. Now, with the existing information, what you do at this point, it is to connect these, to link these with a hypothetical mechanism of action or a known mechanism of action that is the responsible, well, yes, it is the mechanism of action behind the mutagenicity of the epoxide. You build this way a list of structural alerts, and then you use a model, a software that is able to identify these structural alerts, so these features, in your unknown molecule, your target molecule, and this way you have the information related to the possible mechanism of action and the presence or absence of the structural alert in your chemicals. The statistical-based models are based again on known activity. So you will have molecules that are positive and negative. And this is just a very easy representation, a 2D representation of the chemical space, but in reality, you will use lots of different molecular descriptors, and you have to think that we will be in a multi-dimensions chemical space. So you have the molecular descriptors, and you have distribution of the molecules, and the idea again here, it is to group molecules with the same type of activity. But in this case, you're not looking for the chemical function that are in common, but the idea is to build a model that is able to build an equation like this one, where you have the molecular descriptors, and applying this equation, you can predict the toxicity. So in the case of statistical-based models, what you do, if you have your query compound, you calculate your molecular descriptors. Your query compound will have a space, will be projected into these chemical space, and according to the surrounding molecules and their activity, it will be predicted as positive or negative. And typically when we have a binary property like this one, the prediction is a probability of being positive. As in this case, probability of being positive, and so it gives a positive prediction of your target compound. So we have these two types of approaches, and they have different strengths and limits. So the strengths of the rule-based QSARs are that they provide information on the mechanism of action. So it is that you don't have only a prediction, but also rationale behind this prediction. The limits of these approaches are that usually they are not able to explain the difference in the activity within the same chemical class. If you think how these methods are built, we are trying to find functional groups that are responsible for mutagenicity, but it is difficult to consider also the possible mitigating factors of these of the presence of this functional group. So typically, a rule-based QSAR is able to say that a specific functional group can be potential mutagenic and without explaining the difference in the activity in the same chemical class. This is a simplification, of course. I mean, different models can give different information, but typically, this is the type of information that you get from a rule-based QSAR. And there is also the issue of the applicability domain with the rule-based QSAR that is often ill-defined. With the applicability domain concept, it is a concept that tells you if the chemistry of your target structure is described by your model. And when you have a negative prediction by a rule-based QSAR, you don't really know if your chemical is outside the applicability domain, so the chemistry of your compound is not known by the model, or it is really a negative prediction. The statistical-based QSAR, on the other side, they have as strength the fact that they are usually higher. There is usually higher accuracy, so they are able to explain different activities within the same chemical class. So, you know, the limit of one is the strength of the other approach. With the statistical-based QSAR, the definition of the applicability domain is not a problem. Again, strength and limit. But the main limit of the statistical-based QSAR is that the interpretation is difficult because they don't give information about the mechanism of action. But this is given by the rule-based QSAR. So as you can see here, you can use these two approaches together because they can work in synergy, as the strengths of one approach are the limits of the other one. And this is why using these two together, you can improve the reliability of your prediction. And this is above all true for mutagenicity. Now we can have a look at the hazard assessment elements of the guidelines, so the ICHM7 guideline. And what you do is that you apply these two approaches and you evaluate the existing information, and you classify your impurity in one of these 5 classes. 3 are for mutagenic impurities, and the last 2 are for non-mutagenic impurities. And according to this class, you have a different action to control the presence of these impurities. The first 2 classes are assigned according to the existing experimental data, and the last 3 classes can be assigned based with in silico models, so with the QSAR models. This is the workflow, how it works. Once you have identified your impurity, you collect and assess the existing experimental data. If you're able, if these data are enough for a classification, you can classify your impurity as class 1, mutagenic, carcinogenic, or class 5 non-mutagenic, or there is a class 2 that is mutagenic with unknown carcinogenic potential. If the existing data are not enough to come to a conclusion at this point, what you can do is to apply QSAR or generate new testing data, or to use QSAR, two models together, and according to the overall assessment of your predictions, you can predict your impurity as class 5, so it is non-mutagenic, or class 3. It is predicted as mutagenic. So here you have potentially mutagenic impurities, and you can use the class 3 thresholds to control the impurity. Or if you're not happy with that, you can perform a names test and then classify your impurity as class 1, 2, or 5. There is another class here that is called the class 4. And in this case, this is the case where you have prediction that are not in agreement, and using, as an example, a shared structural alert with a non-negative, you can somehow add some additional information to the rule-based QSAR, so that if you have a structural alert, but your structure is very similar with another structure that is negative and has exactly the same structural alert, you can use a class 4. So expert opinion and classify to class 4. So yes, the ambiguous situation can be clarified with an expert review, and you can use your knowledge and additional information to assess, to give an overall assessment based on QSAR prediction. So there exists a lot of different tools that contains models to predict mutagenicity. This is a list of the freely available tools. It's not complete, but these are the best known and the ones that I typically use. And we have a list of the commercial ones. Again, it might be not complete, but this is the well-known and the ones that are typically used. The order is alphabetic order, so they are included in these tables in a pure alphabetic order. Now, which are the features and the positive, the pros of using freely or commercial available models? Well, for the freely available ones, of course, cost is one because they are free, so it's very good, and they are fully transparent in the sense that algorithms and training set are published, available, and so the authorities, they like this fact because they can understand completely what it is your model, which type of information your model is using, and so on. On the other side, the commercial models, they have on their side the fact that typically they have a wider applicability domain, because they are also using, typically what they use are also information that comes directly from pharmaceutical industries, and so they have more information. They have a wider applicability domain. All these commercial tools, they have two approaches implemented in the same tool, in the same environment, while the freely available tools, they usually have either the statistical-based or the rule-based, but they don't have the two together that you can use together. The other pro of the commercial tools, it is that they have tools to generate a report in a much easier way and much quicker than for the freely available tools. And the other thing that you have in the commercial tool that you don't have as easy in the freely available tools are additional information to carry out an expert review. So I'm not saying that these information are not available in the freely available tools. It just requires much more time to find this information there. While these commercial tools are there to give you all the information that you need for your final assessment. And yes, this was the last one. And you have my email address here if you have some questions in future. And thanks a lot for the attention.